Hi everyone, we're going to be talking about DNA replication today, and we're going to be working on skill A, which is looking at models and representations and understanding what they're telling us about processes. This is the note sheet that you should have ready to go, so you want to split your screen so you can see the video and your notes at the same time. Our topic is DNA replication, but the question we really want to answer is how does DNA make exact copies of its code? You should stop the video right now and write this question in the essential questions section of your notes. Replication is a new word for us. It means to make an exact copy. So you will want to stop the video and write this definition at the top part of your notes. A cell needs to replicate its DNA because when it goes through mitosis or cell division, there will be two cells. So we will need to have DNA for cell number 1 as well as DNA for cell number 2, and those two pieces of DNA need to be exactly the same. Remember that DNA has a double helix structure that we can see here. That's so that the bases are tucked to the inside and are protected. But if we need to make a copy, we're going to have to open that ladder up so that we can expose the code. To do this, an unwinder enzyme lands on the DNA and begins to unwind and unzip the DNA. The unwinder enzyme is called helicase because it opens the helix. Pause the video now and fill this in on the notes and diagram. Once the DNA is unwound and unzipped, the DNA code is exposed on both strands. So here is strand number 1, and the code, the A, C, T, etc. are exposed on this side, and on strand number 2, the T, G, A, etc. is exposed on this side. Now the builder enzyme lands on the top of both of the old strands and starts taking floating nucleotides and fitting them into the bases on each strand. You can see that the thymine has been matched with the adenine, so it took a thymine and put it into where it fit with the adenine. And here, down here at the bottom, you can see that this adenine is being fit into the thymine on the old strand. You can also see how the same thing is happening on this strand. This enzyme is called DNA polymerase because it builds a DNA polymer, little pieces building into bigger pieces. Pause the video and write this information on your notes. Once DNA polymerase has finished building the molecules, a team of enzymes move down the new strands to try and find any place where the bases may have been mismatched. Once they find them, they cut them out and fix them. This group of enzymes are called spell checkers. Pause the video and add this information to your notes. So now we have our result, 2 identical DNA molecules. The way that this is done is called semi-conservative replication because you can see on the molecule that the old pieces are on the outside and they are exactly the same pieces that were on our original DNA. But on the new molecules, there is one new strand on each one of them that was built by the DNA polymerase. So having half old and half new means that the molecule has been semi-conserved. Conserve means to hang on to in its original form. Now remember that skill A means that you can look at any diagram and recognize the things that we just saw. So on this diagram, we can see that here is our original DNA in its double helix form. We can see here that what we're not seeing, the helicase, is actually unwinding and unzipping. We can see in this diagram that the DNA polymerase, which is also not shown in the diagram, is beginning to build a new molecule. And in this diagram, we can see that process continuing until we have 2 new molecules that are semi-conservative. We can see the old DNA as the blue part and the gold DNA, which is the new part, half old, half new. Now that you know how replication works, it's time for you to try some practice. So there are practice pages either in your packet or attached to your note sheet, and you should be able to check your answers with the key that is on Schoology. Good luck.